Good morning. At the onset, thanks to the organizers for giving me an opportunity to be here. This is my institute where I started my learning, and it's a pleasure and honor to be heading the department where I joined as a student. I don't think pre-transplant workup is something new. For those who are already in the transplant field, my apologies for the repetitions you may go through. But for those who are new in the field, I would like to just to walk you through as to what the pre-transplant evaluation needs to be done for a solid organ transplant. Of course, we all are aware transplant is going to be a thing that nobody can do alone. It's a big team effort, and lots of specialties get together, and all of them contribute enormously. Adding to that is the transplant immunology specialty, where lots of investigations are required, both pre and post transplant. Do we have a pointer or something? Or anyway, I'll go through this. So the transplant immunology laboratory is supposed to be doing a lot of efforts in the sense that we need to do HLA matching. Of course, we do a mismatch transplant as well, but HLA matching does play an important role. In addition to that, cross-matching, cross-reactive antibody assessment, sensitization, donor-specific antibody, and characterizing the antibody is something that we really need to do if we want to have a successful and a vibrant program. And all specialties get together to make sure that this works fine. So when a solid organ transplant happens, imagine what the immune system is going to do when there is a foreign kidney in the body. Obviously, the immune cells will migrate from the kidney to the lymphoid organs, and that is where the allorecognition happens. That's the initial part. In the very beginning, there is a direct allorecognition of the donor MHC molecules on the surface of the DCs by the recipient T cells. That's what happens in the very beginning. As the process goes on and the graft progresses, there is an indirect allorecognition wherein the immune cells migrate to the kidney, donor kidney, grasp the cells, and grasp the antigen, and then again back to the lymphoid organ where the antigen presentation happens. So both this direct indirect mechanism enhances the immune responses, and that's why we need to really keep a check on all these immune cells when they are moving. If we have a mismatch graph, obviously there's going to be a heightened immune response, and you all many of you are aware that there's lymphocyte depletion, antibody depletion to be done, and of course we need to keep in mind the unacceptable antigens that if they are there, what do we need to do and how do we handle them? And antibodies definitely are there because of pregnancies, transfusions, and if there is a retransplant, the prior organ that has gone in also leads to antibody development. So at various levels, all these immune cells need to be kept in check, and there are several products that are helpful to keep this immune system under control and suppress this so that there is no infiltration of the alloreactive cells and the kidney is not damaged. And basically, the better the matching, the better control we have, and lesser the immunosuppression required so that we can avoid all of these mechanisms. And what we need to do is everybody wants to just jump over this hurdle that we don't have rejection. And if there is any acute or chronic, how do we counter that? Or maybe develop biomarkers that we can predict these rejection might happen and then to act preemptively. So the main things that we need to keep in mind, HLA matching between the recipient and donor. Of course, you all may disagree that no, there's a lot of mismatching and the transplant still does well. Does matching plays a important role in long-term survival. Donor-specific antibodies, if the recipient are sensitized, we need to monitor what these antibodies are, characterize them, and human-to-human variability is there in drug absorption and clearance. And in this part of the world, infections are something that we all need to handle no matter what we do. So in any transplant immunology laboratory, what we basically do is find out if there is an HLA match, good. If there isn't, what is acceptable? Then we have to do genetic relationship establishment. Many of the people do with HLA typing, but I would like to highlight that as per the TOHA Act, HLA is not the way that we need to establish genetic matching. Relationship, cross-matching, different protocols, CDC and its modification, and flow cytometry. We have an expert, Dr. Handu is here. He's going to talk to us. Sensitization status, we need to screen the antibodies or do a PRA assessment or do a, donor-specific antibody assessment, and that's what we need to keep in mind. When we are characterizing antibodies, to which locus, what's the cross-reactivity, whether they're complement fixing, C3d binding, what type of IgG, whether the antibody is persistent or transient is what impacts the graft. And then the strength of the antibody, meaning MFIs, do we keep in mind MFIs or not, is what we need to see. HLA matching, many of you will be aware, we look at the class 1, A, B, C, and the DR, DQ, and the DPs. and it's on the chromosome 6. Of course, a very, very polymorphic system, very polygenic, and important immunologically relevant antigens being present in this region, therefore very crucial role in any transplant program, whether it is solid organ or stem cell transplant program. So just imagine if we have, all of us have a pair of chromosomes. So if we have a homozygous locus, every locus is homozygous, these glycoproteins on the surface will be same. So we'll have very minimal 1,1 HLA molecule on the cell surface. Whereas if we have a heterozygous individual, heterozygous sequences at all the loci, an individual can have as a max of 18 HLA alleles on the cell surface. So these are glycoproteins on the surface. So imagine if we have a unrelated individual or a relative also, then the differences could be a lot in the HLA. Of course, when we have the family, it's going to be less. So this is just a graph indicating that the development of technology is helping us identify lots and lots of genes. So it's highly, highly polymorphic system. At the moment, 42,000 HLA genes are known, HLA alleles are known, and a laboratory needs to be competent enough to handle all this polymorphism, which lies in the peptide binding groove, and that's how the immune response is handled. And this is the way we put down the HLA. That's the nomenclature of HLA. If we have done molecular assays, the HLA, the locus, the gene, and then further on characterizing the coding and the non-coding sequences if we have run the next-gen sequencing technology. This is a chapter that me and Ajay had written in the book released by Dr. Vinan Bhargava and team from Gangaram, and there all the methodologies have been listed out. So any laboratory that needs to do HLA typing would definitely have all of these protocols available, whether at the moment practicing SSP or not, but the laboratory starting up would always prefer to do the single sequence specific primer methodology, which is like a low throughput. You can do only a few samples in a day. Whereas if you have a Luminex based or a sequencer or a next gen sequencer, the throughput is much higher, resolution is much higher, and all the transplant immunology laboratories do use this platform. So Luminex is a platform where we use fluorochrome conjugated beads, and then we can use this for HLA typing as well as for antibody assessment. And many of the nephrologists or the surgeons who are in the field would be interacting with the labs and hearing about these. So usually every transplant immunology laboratory would start from serology, at least for the CDC cross-match, the serology is to be done, and move on forward to the next gen sequencing, and now the third gen sequencing has also come up. So the possibility of newer technologies coming up, and people have to keep up with the pace of having these technologies available in the lab. So when we are looking at a family, when we have a pair of chromosome 6 parents, two parents, and how the genes are inherited is one chromosome paternal and one maternal. The very characteristic feature of HLA is that it is inherited en bloc. So if you have an HLA A, B, C, DR, DQ, all of the genes will be, they are very tightly linked. So they're inherited en bloc. So basically we have only these four combinations that anybody can inherit from a parent. So if a child is born with a B and a C combination or any one of these, there's a 25% chance that one would find a full match sibling in the family, and there's a 50% probability that one would find a haplomatch, I mean one chromosome sharing from the parents is inherited, and there's equally 25% chance that they would be completely unidentical. So that's how the HLA inheritance is, and this is probably the reason that people thought that we could use this for establishing genetic relationship. But unfortunately for solid organ transplant, as per the THOA Act, this is not, because when we look at the population, there's a lot of diversity in population, but we do have so many common genes that if I test all of you among the hall, I probably will find maybe 10% of you matching half the genes with me. So it doesn't mean that I'm related to you. It's just that those common genes are available in a particular population, and we may have a 50% sharing. So going by HLA typing for genetic relationship is not fair and not to be done, and that is what the THOA Act actually mentioned. In 2008, the Act did mention that the HLA genes could be used to establish relationship between the near relatives. Of course, the Act says that the authorization committees need to look into the documentation, and there are specific documents to be looked into. And it also stated that results of tissue typing need to be looked into to be establishing the relationship. And then the HLA was mentioned at that time, just the HLA-B and the DR by molecular methods. However, because of the understanding that two unrelated individuals can also have similar match, not only 50%, even 100%. Therefore, the Act was modified. This Form 3 that was being used earlier had mentioned that based on the products of human MHC, that one could establish the relationship, but the same thing has been changed when the act was modified in 2014. And this act now states that if these documents are not satisfactory, and if the competent authority finds that the relationship is not conclusively established, then the test needs to be done is the DNA profiling, which is very different from what the HLA typing is. And this needs to be done from a laboratory that is accredited by the National Accreditation Board of Laboratories in India. So this is very important fact, and then only the person should be signing the Form 5, because it very clearly now replaces the HLA MHC with DNA profiling. So many of the centers are asking their laboratories to provide the reports on the basis of relationship, on the basis of HLA. I think that's unfair, and that's deviating from the statutory guidelines. We shouldn't be doing the HLA. HLA is important for biological understanding, for the matching, and also for understanding of the donor-specific antibody. So that is where the clinical relevance of testing HLA and looking at the genes is not for establishing relationship. Of course, when we do parents, grandparents, we do find the inheritance, but I think the Act doesn't permit us to use the HLA for doing so. I'm sorry about this slide. This was something talking about the paper in 1969, NEJM, where Sir Paul Terasaki actually identified that looking at a cohort where there were cross-match positivity, higher number of patients had rejection, and that's where the transplant immunology field started off with his paper in NEJM, that HLA typing could be done by CDC and we could do a cross-match. So the cross-match is very important, where we basically use this plate which was designed by Paul Terasaki. So this is the Terasaki plate where we have the recipient serum, we add donor lymphocytes, add the complement, and wherever there is a complex formation, it indicates that the recipient serum does have antibodies to the donor HLA or donor antigens, and that's when we see the positivity, and the cross-match positive is a contraindication for a transplant. So if we use eosin formalin dye, this is how the plate under the phase contrast microscope looks like, and that's how we need to identify the cross-matching. And then we have Luminex-based methods, we have sequencing, the Sanger sequencing, and the next-gen sequencing. In the next-gen sequencing, the whole idea is that we can fragment the DNA into several parts and sequence it 100 times so that we know exact sequence after doing the next-gen sequencing. In Sanger, we may still have some ambiguities, which is not very good to have. So our laboratory did do the NGS-based established technologies, and when we studied several populations, we could identify several novel alleles. So sometimes we do feel very, pathetic in the sense that if there is a patient who has these novel alleles and we don't have the beads on the antibody profile where we need to have testing, then we may miss sometimes the antibodies to those. So these novel genes are there. We've published many of them, and no matter whatever the technology is available, we still carry on with the transplant. So when we are looking at the donor, ideally we want a perfect match. But of course, if the donor is homozygous for HLA-A or HLA-B, it's a zero mismatch. So the concept has changed from match to mismatch. The zero mismatch is equally good. But if we have mismatches at either 1 or 2 or 3 loci or all the loci, that is the worst. So that's the criteria for a best to a worst match. And that's what we look for. But when we are looking at mismatches, this is another figure which tells you if this is the recipient, all the 6 genes mentioned, if the donor is homozygous at 1, it still is 3 match but a 0 mismatch. If there's a matching for all the 5, there's only 1 locus homozygous, still 5 matching but a 0 mismatch. And similarly here 6 match and 0. So the 0 mismatch is what we need to look into. The mismatches would lead to antibodies. And this is a very old graph from 1990 to 2014 indicating a collaborative transplant study that a 0 mismatch survival is definitely much better than 6 mismatch. I haven't changed it to any current graph because the fact is the same, that the mismatches are going to definitely impact the survival of the graph. And also the long term, if you see that the 0 to 1 mismatches, the long term survival is much better than the green graphs, and the red graphs is the one which is 4 to 6 mismatches. Even the rejections are higher. I'll be quick. So the antibodies are developed in majority of the cases. About 25% do develop antibodies. We all know the reasons for the antibodies. And definitely the cross-match assays are being done, different cross-matches just to check the antibodies. But the cross-match has to be also looking into whether it's a functionally relevant assay or a non-functional assay or just a binding assay. So CDC is a functional assay because here we can look at the reactivity between the recipient antibody and the donor cells, whereas when we do the flow cytometry, it's a binding assay. It's not a functional assay. Of course, its sensitivity is much higher. Single antigen bead assay is also a binding assay, but if we add into that the complement binding, then it also becomes a functional assay. So we all have to see when and what test has to be utilized for the purpose that we're wanting to. So binding means single antigen beads or flow cross match, and the functional assays are when we do the CDC and the complement binding. Of course, flow cytometry, Dr. Hando is going to talk to you. These antibodies can act in different ways, acting the complement or the ADCC or opsonization or activation of the endothelial cells. So several ways that these antibodies can harm the graft. So this is just an example that
I'm
we did only a
extremely
few
sorry to
loci testing.
intervene, ma'am. I request you to please
I'll
sum up
quick.
in 30
So,
seconds.
okay. If we do just a few loci testing, then we may not be able to characterize the antibody. So the all the loci testing is really important for monitoring a graft. Of course, there are papers to indicate that when the MFI values are a certain level, for example, in this publication says when the MFI levels are more than 7,000, the CDC crossmatch was positive. We didn't have to do in the lab. When the MFI values are more than 3,000, then the flow cytometry crossmatch was also positive. So basically the binding between the structural epitope and the paratope is what we need to look at. And there are functional residues that are very important. So if we have all of these three functional residues, then the antibody would bind to the epitope. And then so the cross-reactive antibodies that we need to keep in mind when we are doing the antibody assessment. Okay. So just that when we do the antibody assessment, we need to check whether the antibodies are transient or persistent, whether they are preformed antibodies, to what locus, whether they are IgG, IgM, or which class of antibody, then C1q fixing, C3d binding, because they do impact the graft, as we see in many publications, the graft survival, and also depending on the preformed antibody. If they clear up, it's good, and also the antibodies that bind to the complement C1q, they don't clear off very easily, so they are harmful for the graft. And now the options have come up. We are not only looking at the HLA alleles, we can look at the epitopes and the aplets. And these are very important, that if there is an aplet mismatch, that's going to be important to predict whether that's going to be a graft outcome is going to be good or bad. These are several aplets known. And HLA match may be similar,
I
but the
am
aplets can be
extremely sorry,
different.
ma'am. We have other speakers waiting.
Okay.
Thank you.
Thank you very much for your time.